Hello, everyone. Welcome to our presentation. My name is Dr. Jeremy Petrovich, editor at Wiley's Current Protocols. It is my pleasure today to introduce our two speakers for this virtual seminar titled Advancing Human-Induced Pluripotent Stem Cell-Derived Blood-Brain Barrier Models for Studying Immune Cell Interactions. We are joined by Dr. Britta Engelhardt of the University of Bern, Switzerland, and Dr. Hideki Nishihara of the Yamaguchi University. Dr. Engelhardt is a world-leading expert in brain barriers research, with a focus on the role of the brain barriers in controlling immune cell trafficking into the central nervous system. Dr. Nishihara's research focuses on the blood-brain barrier with a special emphasis on the pathomechanism of neuroimmunological diseases. Together, they have established a novel protocol to differentiate blood-brain barrier models from human-induced pluripotent stem cells applicable for neuroimmunological diseases that they will share with us today. Please welcome our speakers and remember to remain for the question and answer session that follows the presentation. Thank you.
Welcome to this seminar on advancing human-induced pluripotent stem cell-derived blood-brain barrier models for studying immune cell interactions. My name is Britta Engelhardt, and I will be giving the seminar together with Dr. Hideki Nishihara, who will follow and tell you all the details about these models after my introduction. So first of all, I would like to introduce to you the blood-brain barrier, and what you see here on the left, this is established at central nervous system microvessels at the capillary, but also the post-capillary venule level. And the blood-brain barrier itself is at the level of these endothelial cells that are highly specialized. And then they are embedded or surrounded by a high number of pericytes embedded in the basement membrane of the endothelial cells. And then in addition, astrocytes that process their entry towards the vessels and establish an additional basement membrane, the parenchymal basement membrane, surround these vessels by a structure called glia limitans. Now, the continuous crosstalk of these pericytes and the astrocytes with the endothelial cells maintain their unique properties. This is very important. The endothelial cells themselves establish a physical barrier because of very complex tight junctions that are highlighted here. They prohibit the free diffusion of molecules via the paracellular pathway. And then there is also a physical barrier through the endothelial cell proper because they have a very low pinocytotic activity, so there is no free transport of molecules through the endothelial cells. Now, having said that, once you start to listen to me now and your neurons need sugar and energy, it's clear that, of course, nutrients have to get into the CNS and toxic metabolites have to get out of the CNS. To this end, these endothelial cells are biochemically unique by expressing transporters and efflux pumps that are not expressed by endothelial cells elsewhere. Last but not least, at the level of the post-capillary venules, these highly specialized endothelial cells also control immune cell entry into the CNS. Now, you may know that in multiple sclerosis, which is considered an autoimmune disease of the central nervous system, the early pathological hallmarks are, in fact, blood-brain barrier breakdown. So this is used as a diagnostic criteria, which you see here in the MRI imaging, highlighting here these gadolinium-enhancing lesions, meaning that gadolinium can diffuse across the broken blood-brain barrier into the tissue. You can also see, if you have access to the tissue, that immunoglobulins staining here the whole tissue diffuse across the defect blood-brain barrier. And then eventually you have immune cell infiltration into the CNS. You see here a blood vessel, and all these brown cells have infiltrated into the CNS where they usually don't belong. And this eventually leads to demyelination, as you can see here by these white areas in the tissue staining for myelin, you're lacking myelin. Now, we can model multiple sclerosis in animals, and there's a disease that's called experimental autoimmune encephalomyelitis, EAE, and EAE actually is a very good model for the very early hallmarks of MS, and again shows the same, namely immune cell infiltration into the CNS and blood-brain barrier breakdown. Now, this looks as follows. You see here in red CD45 immune cells that have crossed the endothelial and the parenchymal basement membrane, so they are in the tissue parenchyma. And this is accompanied by leakiness of the blood-brain barrier, which you see here by this orangey staining, which is a staining for fibrinogen, surrounding here again an inflammatory cuff. At this time, the animals develop a clinical disease, having a limb tail, becoming paraplegic. And if you look exactly where we are, as I told you before, you can see here that the immune cells have crossed the endothelial basement membrane, depicted here in green, here it is yellow, and then the second basement membrane of the glia limitans, and have reached all the way off the CNS parenchyma. This causes the disease. Now, the general idea usually is that in multiple sclerosis, and this depicts now from the healthy to the disease state, what you see here, you have these perivascular infiltrates, eventually reaching into the parenchyma, upregulation of adhesion molecules, leakiness of the blood-brain barrier, that this is a consequence of the immune cell infiltration into the CNS. But we have questioned that. Could it be that a breakdown of the blood-brain barrier or change in the blood-brain barrier is not a consequence of neuroinflammation, but contributes to the disease? Now, it's very difficult to really study this because we have very easy access to immune cells from our patients, which allows us to investigate their contribution to MS pathogenesis. However, it's not necessary to make brain biopsies in order to diagnose MS. So the lack of access to brain tissue from MS patients, and thus really cells of the blood-brain barrier, hampers investigating the contribution of the blood-brain barrier to MS pathogenesis. Now, there is a solution for this more recently, and this is that we can now start to disease model using human-induced pluripotent stem cells. They have proven to be a scalable and renewable source of cells from one individual and have been differentiated into numerous different cells from the central nervous system, amongst those also other brain cells, the endothelial cells. So at this point, we had the idea to team up with two different groups in order to study if we establish from human-induced pluripotent stem cells blood-brain barrier models from healthy controls in MS patients, if we can identify maybe changes in the blood-brain barrier from these patients. So to this end, we started a collaboration with the group from Renaud Dupaquier from the CHUV in Lausanne in Switzerland, who has established human-induced pluripotent stem cells from MS patients and healthy controls. And a second group, and this is from Eric Schuster, who have established numerous blood-brain barrier models from human-induced pluripotent stem cells. And the whole collaboration started to become productive when Dr. Hideaki Nishihara joined us from Yamaguchi University as a postdoctoral fellow and spent 4 years with us here at the University of Bern. And with this, I will hand over the word to him, and he will describe you his journey to Madison, Wisconsin, and how we started to establish and advance human-induced pluripotent stem cell-derived blood-brain barrier models together that we can now study immune cell interactions and brain barrier defects in MS.
My name is Hideaki Nishihara. I guide you through how we recognize the importance of establishment of new model and how we develop new methods, and then how we use for studying pathogenesis of neuroimmunological diseases. When I started this project 3 years ago, there are already two methodologies available to differentiate iPSCs into brain microvascular endothelial cell-like cells, BMEC-like cells. First method is reported by Liebman in 2012. We call this method unconditioned medium method, since this method uses unconditioned medium to co-differentiate with endothelial and neural precursor cells, and then purified BMEC-like cells using endothelial cell-specific basement membrane. This method is most widely used and has since been adapted to accelerate endothelial maturation by optimizing seeding density or by improving the culture medium. Second method is reported by Kian in 2017. This method is called defined medium method, since this method uses chemically defined medium. BMEC-like cell is induced by sequential activation of Wnt and retinoic acid signaling. We first tried protocol called DM method because we think defined medium gives us less variable results and good reproducibility. This slide shows our strategy, how we study patient-derived BBB-forming endothelial cells. In our case, we took erythroblast from healthy control and MS patients and reprogrammed them into iPSCs using nucleofection with episomal plasmid, and then redifferentiated into brain microvascular endothelial cell-like cells using DM method. Using DM method, we found that both healthy control and MS-derived BMEC-like cells are positive for tight junction-associated protein Z01, as you can see here in the top line, and then tight junction protein claudin 5 and occludin. However, the cell size is larger in case of MS compared to healthy control. We also noticed that staining of tight junction is straight, and sometimes junctional staining cross over the nuclei. We next studied the diffusion barrier function of this BBB model by checking transendothelial electric resistance, shortly TER. In this case, we use electrode and check the resistance between upper and lower wells, and usually higher resistance shows the tight barrier. Both healthy control and MS showed extremely high TR value, range of several thousand. This value is similar as reported in vivo TR. When we compare MS versus healthy control, we found that TR values is low in MS compared to healthy control. We also performed permeability assay to confirm these findings. In this case, we put sodium fluorescein into the upper wells and wait 60 minutes and collect sample from bottom well to measure how much molecule passed through the DMM BMEC-like cells. Again, we saw that MS-derived DMM BMEC-like cells has higher permeability, meaning MS-derived cells is leaky compared to healthy control. These cells also showed functional expression of efflux pump. We did PGP accumulation tests using PGP substrate rhodamin 1,2,3 and PGP inhibitor cyclosporin A. Addition of cyclosporin A inhibit efflux pump activity and increased intracellular accumulation of rhodamin 1,2,3. Both MS and healthy control derived DMM BMEC-like cells have functional PGP since rhodamin 1,2,3 accumulate upon cyclosporin A treatment. However, efflux pump activity is also low in MS cases compared to healthy control. We next checked endothelial adhesion molecules, which is important when immune cells interact with the BBB-forming endothelial cells. DMM BMEC-like cells shows ICAM1 under non-stimulated condition. Also, ICAM1 is upregulated upon TNF-alpha and interferon-gamma treatment. When we compare healthy control versus MS, we found that MS-derived DMM BMEC-like cells shows enhanced cell surface ICAM1 staining under both non-stimulated and stimulated conditions. Therefore, we confirmed that conventional DMM BMEC-like cells show extremely tight diffusion barrier and good efflux pump expressions. And we saw difference between MS and healthy control with respect to diffusion barrier characteristics, efflux pump activity, and ICAM1 expressions. At the same time, we noticed two issues of this conventional method. First, these cells show mixed endothelial and epithelial phenotype, as you can see tall nuclei and straight junctional protein like epithelial cells. This means that we cannot use these cells as a simple BBB model. Second, These cells do not express proper addition molecule including VCAM1, which is upregulated in brain endothelium in MS. You may note that this VCAM1 is indirectly therapeutic target for one of the most effective treatments for MS, called natalizumab. Therefore, we thought that we cannot use conventional method to study MS pathogenesis, and started to establish completely novel method. Before I explain our novel method, let me introduce you to sequential extravasation cascade when immune cells cross blood-brain barrier forming endothelial cells. As Britta introduced you to BBB function at the very beginning, BBB controls immune cell entry into the CNS. Unfortunately, this part is usually not well considered when we talk about BBB model. However, when we think about neuroimmunological disease or neuroinfection, as well as tumor metastasis, immune cell infiltration is very important. When immune cells from the peripheral blood cross the blood-brain barrier in the serum, they first need to slow down their speed, and this step is called capture or rolling. This is mainly mediated by selectin. Next, immune cells are activated via chemokines, and they change conformation of integrin, and then firmly adhered with endothelial cells using VLA4 VCAM1 and LFA1 ICAM1 interaction. One BBB specific phenomenon is that immune cells crawl on top of endothelial cells against the direction of the flow to find good place to go through endothelial cells, and then cross endothelial cells, and finally reach the CNS parenchyma. This slide summarizes well-known endothelial adhesion molecules which mediate sequential extravasation cascade. What I want to highlight here is that if we wish to study immune cell entry within the CNS, we need to use models which possess all these markers. And if we go back to conventional DMM BMEC-like cells, these cells express ICAM1, CD99, and PCAM1. However, unfortunately, they do not express ICAM2, VCAM1, P or E selectin. We further investigated expression of UMM BMEC-like cells, which is the first iPS-derived BB model established by Lippmann. The expression patterns of adhesion molecules are exactly the same as DMM BMEC-like cells. We were thinking for several months how we managed to induce VCAM1 using this conventional method. We tried to remove retinoic acid from the protocol. We tried to co-culture with astrocyte or parasite from several different sources. However, we could not manage to induce VCAM1 expressions. Therefore, we decided to establish completely novel method. To this end, we took the advantage of our previous human cord blood stem cell derived BBB model, namely brain-like endothelial cells, BLECs. This model is derived from not induced pluripotent stem cells, but derived from cord blood stem cells. In this case, cord blood stem cells are first differentiated into endothelial progenitor cells, and then induce various properties by co-culture with parasite. And we have previously shown VCAM1 expression of BLECs upon TNF-alpha and interferon gamma stimulation. Therefore, we first differentiated iPS into EC progenitor by activating Wnt signaling, and after 5 days of differentiation, we purified CD31 positive endothelial progenitors by magnetic activated cell sorting. If we checked adhesion molecule expression by flow cytometer, for the first time, we could see the staining of VCAM1 on iPS-derived endothelial cells. Then, we seeded these cells onto the filter insert. However, we noticed that EC progenitors can be differentiated to V-cadherin positive endothelial cells and negative iron, and most of these V-cadherin negative cells are positive for smooth muscle actin. Therefore, we call these cells smooth muscle-like cells. We next try to purify EC from this mixed population. By carefully observing these cells under microscope when we passage cells, we noticed that endothelial cells detach earlier compared to smooth muscle-like cells. Therefore, we selectively passage early detached endothelial cells several times and could obtain pure endothelial cell layers. If we checked adherence and tight junction proteins by immunocytochemistry, these cells developed tight junction protein claudin 5 and occludin after several passages. This development of tight junction proteins are accompanied by functional outcome. Although passage 1 cell does not show any TL values, and we found extremely high permeability value for small molecule tracers. Only one passage is enough to show good barrier properties. TL value is range of 60 to 80 ohms per square centimeter, and permeability value for sodium fluorescent is below 0.3. These diffusion barrier properties are comparable or even tighter compared to primary microvascular endothelial cells. Since these cells express important tight junction proteins and good barrier properties as detected by moderate permeability and TL values, we term these novel method-derived cells as extended endothelial cell culture method-derived brain microvascular endothelial-like cells, shortly EECM-BMEC-like cells. And as you can see here, the morphology of the EECM-BMEC-like cells are completely different from DMM-BMEC-like cells. EECM-BMEC-like cells express mature zigzaggy shape of junctional molecules, flat nuclei, as primary brain-forming endothelial cells. We could obtain good barrier properties by extended cell culture and passaging. However, we again faced Wickerman expression issues. As you can see here in this histogram of adhesion molecule staining by flow cytometer, Wickerman staining is reduced in EECM-BMEC-like cell monoculture compared to naive ECs. Induction of Wickerman in EECM-BMEC-like cells was the most difficult part when we established this method. We were again thinking several months how we overcome these issues. At the beginning of development of this method, Wickerman expression was sometimes positive and sometimes not. We would regularly discuss with Eric Schuster's team, and we noticed the difference of contamination of smooth muscle-like cell is key of expression of Wickerman. In other words, if we had smooth muscle-like cell contamination in the same dish like left picture, we saw high Wickerman expression. And if we don't have smooth muscle-like cell contamination, Wickerman expression is very low. Therefore, we decided to co-culture smooth muscle-like cells with EECM-BMEC-like cells. As we expected, we could obtain positive Wickerman staining on EECM-BMEC-like cells by co-culturing with smooth muscle-like cells. We further found that smooth muscle-like cell co-culture could be replaced by conditioned medium from smooth muscle-like cells. These data indicates that humoral factor from smooth muscle-like cell induced expression of Wickerman on EECM-BMEC-like cells. And this finding is confirmed using several donors and semi-quantification by delta mean fluorescence intensity. We also found that conditioned treatment is working not only from abdominal side, but also from luminal side, which allow us to make easy setup of immune cell interaction assays. We finally obtained good addition molecule expression including ICAM1, VCAM1, ICAM2 or selectin as observed in vivo by co-culturing ECM-BMEC-like cell with smooth muscle-like cells. Furthermore, we confirmed functionality of endothelial ICAM1 and VCAM1 by addition assay using blocking antibody against endothelial ICAM1 or VCAM1 or respective immune cell integrin beta-2 or alpha-4. As you can see here in this right graph, addition of anti-beta-2 integrin on T cells or anti-ICAM1 on ECM-BMEC-like cells reduced the number of adhered T cells onto the ECM-BMEC-like cells. With a lesser extent, blocking antibody against alpha-4 integrin or VCAM1 is also working. I would like to show you our entire protocol again and highlight here that smooth muscle-like cells we needed for induction of VCAM1 are byproduct generated during the ECM-BMEC-like cell differentiation and as such do not require independent differentiation of multiple cell types in parallel and are derived from the same iPSC cell lines as the ECM-BMEC-like cells, which should make this system readily applied to isogenic disease modeling. We further checked cell identity of our ECM-BMEC-like cells using RNA sequencing data and found that ECM-BMEC-like cells showed high expression of endothelial cell genes like CDH5, CD34, PCAM1. We also confirmed that ECM-BMEC-like cells express least epithelial cell genes like CDH1, EPCAM, Claudin1, nor Claudin3. We also confirmed extended culture led to upregulation of Claudin5, consistent with previously mentioned protein level observation in ECM-BMEC-like cells. This data reflecting the definitive endothelial nature of ECM-BMEC-like cells. Here, I summarize our novel method ECM-BMEC-like cells. ECM-BMEC-like cells shows highly endothelial phenotype and mature junctional proteins. Furthermore, proper functional adhesion molecule could be induced upon co-culture of smooth muscle-like cells, which is byproduct of the same protocol. Now we have tools to investigate the contribution of BBB-forming endothelial cells for MS pathogenesis. We use this novel method and compare healthy control versus MS BB model. We first investigated molecules related to serum protein leakage. Both MS and healthy control derived ECM-BMEC-like cells stained positive for V-cadherin. However, the size is larger in MS clones, as we saw in previous DM-BMEC-like cells. When we investigated tight junction protein, we are excited to find discontinuation of Claudin5 and occludin in MS cases, as you can see here in these yellow arrows. We are very excited because this is the phenotype of what we see in vivo in MS regions. One of the advantages of our method is that we can analyze function of BBB. Discontinuation of tight junction protein in MS is accompanied by functional outcome as TR value is lower in MS cases, as well as higher permeability value for sodium fluorescein. This indicates that MS-derived BBB is leaky compared to healthy control. We next investigated molecules related to second BBB dysfunction hallmark, immune cell infiltration. We found that MS clone shows pronounced ICAM1 and VCAM1 staining under stimulated conditions. This finding is again the phenocopy of in vivo MS regions. We confirmed enhanced staining of adhesion molecules using flow cyte meta-analysis. And in this case, we could also see the pronounced staining of VCAM1 under non-stimulated conditions in MS. We next investigated the function of this enhanced staining of ICAM1 and VCAM1. To this end, we did adhesion assays using allogeneic Th1 star cells from healthy control. We cultivated ECM BMEC-like cells from healthy control or MS using chamber slide and put allogeneic Th1 star cells from completely different donor and let them adhere 30 minutes. After 30 minutes of incubation, Th1 star cells were washed out, and then remaining adhered cells were automatically counting. The results showed that allogeneic Th1 star cells adhere more to MS-derived ECM BMEC-like cells compared to healthy control, under both non-stimulated and inflammatory cytokine-stimulated conditions. We went on further to investigate not only adherence, but also different behavior of immune cell interaction with BBB in vitro. To do so, we adapted in vitro flow assay from our previous mouse model. We cultured ECM BMEC-like cells using dish, and we perfused fluorescently labeled T cells on top of ECM BMEC-like cells under physiological flow using automated syringe pump. Time-lapse video was made to observe different T cell interaction with BBB in vitro. As you can see here in this zoom-in video, we could observe different behaviors like diabetes as marked in blue, or chlorine marked in orange, or probing in pink, meaning stay in the same position and send protrusions. We could observe different T cell behaviors and conclude that our novel method is suitable to study immune cell interaction with BBB in vitro. We applied this in vitro flow assay using ECM BMEC-like cells from healthy control and MS and perfused allogeneic Th1 star cells. And as you can see here in this video, in case of MS, you see higher number of Th1 star cells adhere to BBB and serum. When we further investigate in-depth post-arrest behaviors, we found that more allogeneic Th1 star cells diabetes across the BBB model in MS cases. Here is the quantification. In the setting of MS, increased number of Th1 star cells adhere and diabetes across ECM BMEC-like cells. If we check the post-arrest behavior, more events in probing or chlorine with diabetes and less detachment events in MS BBB, meaning MS-derived BBB has pro-inflammatory phenotype and actively contributing immune cell migration across the BBB. One of the strong points of our model is that we can investigate autologous immune cell trafficking using immune cell and BBB model from same individual. We finally confirmed our concept by perfusing fluorescently labeled PBMCs from MS or healthy control on top of autologous ECM BMEC-like cells. And as you can see here in this video, in the case of MS, again we see higher number of PBMCs adhere and migrate through BBB and serum. By counting adhered and diabetes autologous PBMCs, we confirmed our findings that MS-derived BBB has pro-inflammatory phenotype and actively contributing immune cell migration across the BBB. Here, I summarize what we found in MS versus healthy control-derived ECM BMEC-like cells. Our method can phenocopy and serial cell changes in vivo in MS brain regions in terms of BBB dysfunction as shown by discontinuation of tight junction protein, and the BBB pro-inflammatory phenotype as shown by pronounced staining of adhesion molecules. Furthermore, we used our in vitro BBB model to analyze function of the BBB and found that MS-derived BBB is leaky and recruit more immune cells into the CNS. From this data, we conclude that BBB dysfunction found in MS is not just the sequencing of CNS information, but rather actively contributing MS pathogenesis. We next aimed to find genes responsible for this BBB disruptive phenotype. To this end, we did RNA sequencing of our models, and we found more than 400 genes differentially expressed between MS and health control-derived EEC and B-microarray cells. These genes include some genes that are important for BBB development and maintenance. One of these genes relates to Wnt beta-catenin signaling. It is widely known that if Wnt signaling is on, beta-catenin translocate into nuclei and promote messenger RNA synthesis of target gene, including claudin 5, which increase BBB function. In MS-derived BBB model, we found that messenger RNA expression of canonical Wnt target gene axin-2 is decreased, and negative regulator FZD6 is increased in MS. This data indicates that canonical Wnt beta-catenin signaling is impaired in MS-derived BBB model. Therefore, we aim to rescue MS-BBB phenotype by activating Wnt beta-catenin signaling. Our strategy is using GSK3 inhibitor, which stabilizes intracellular beta-catenin and induce beta-catenin signaling. Here is the result. By activating Wnt beta-catenin signaling, disrupted claudin 5 staining is recovered on MS-derived BBB model. This is functionally related, as you can see here, decreased permeability to sodium fluorescein upon Wnt beta-catenin signaling activation. And this recovered permeability is comparable to healthy control level. Furthermore, activation of Wnt beta-catenin signaling reduces Beta-catenin expression in MS-derived BBB model. This finding highlights that iPSC's derived BBB model can be used as a tool to identify novel therapeutic target for BBB stabilization. Today, we presented a novel protocol for iPS-derived BBB model. The novelty of our model is pure endothelial phenotype and expression of proper adhesion molecule, including beta-catenin, and thus suitable to study neuroimmunological diseases. Novel BBB model can phenocopy BBB disruption found in MS autopsy samples. In addition, this model has advantage that we can analyze BBB function. In this part, we showed that MS-derived BBB is leaky and attract more immune cells infiltration into the CNS in MS, and conclude that BBB dysfunction in MS do not merely reflect CNS inflammation, but are actively involved in MS pathogenesis. Furthermore, this model is suitable for developing therapeutic strategy for BBB stabilization. As we showed, Wnt beta-catenin activation restored BBB disruption. This slide shows lab member when I worked in Britta Engelhardt's lab in Switzerland and grant support for this project. And I hope you enjoyed this talk and many researchers is interested in our novel method. And thank you for your attention. I'm very happy to take any questions.
Thank you very much, Dr. Engelhardt and Dr. Nishihara, for your presentation. That was a very engaging overview of the blood-brain barrier and the research that you've conducted. So some of the questions we have from the audience, our first one is for Hideki, if VGEF increases mRNA in blood plasma, does it cause pathogen modification or regeneration process modification?
Thank you for this question. I guess this question means that if VGF is causable molecule or necessary for regeneration of BV damage. So I think VGF have both role, but more causable because recently it is shown that polymorphism of VGF is related to MS susceptibility. And also, second, we have previously shown that if we incubate immortalized human BV model with MS serum, brain microvascular endothelial cells secrete VGF and then disrupt BV model by decreasing the tight junction protein. So I think VGF have both role, but they're more causable for MS pathogenesis.
Great, thank you. Our next question also is, Hideki, I think be directed towards you, is fibronectin can act to trigger neuroinflammation. What do you think about a model without the use of fibronectin, and may that negatively affect the success of the model system?
Yes, thank you again. And so in our model, fibronectin use for the increase the adherence of the cells onto the filters. So we generally use fibronectin only for the filter, so transfer filter made by polycarbonate. And when we use, for example, plate, dishes, or glasses, we only use collagen. But of course, fibronectin is have a pro-inflammatory phenotypes. But to increase adherence, we need fibronectin. So unfortunately, we didn't have a comparison between fibronectin positive or negative coding. But if we don't use the fibronectin, the cells doesn't grow onto the polycarbonate filters.
Thank you. Next question, Dr. Englehart, would be for you. The audience member first, thanks you both very much for the nice presentation. And does the EECM BMEC cells also form tubes in a standard tube formation assay?
Mm-hmm. That's a very nice question. Thank you. And the disappointing answer for you is we don't know yet. It's actually Ethan Lippman is trying this with the different differentiation protocols that Hideaki has introduced before, which we refer to as a defined or undefined method. And they can form tubes, these endothelial cells derived from stem cells, in different modified 3D environments. So I'm pretty positive that also our model would do this, but we have not tried this ourselves.
Thank you. Hideki, I believe you may have touched on this, but just one of the audience members was curious, what type of support did you use to differentiate stem cells into BM and BMEC-like cells?
Yes, thank you very much. And in our case, in this method, we use growth factor reduced matrigel. But recently, we have a trouble of issue of the delivery of matrigel. So now we are also trying to use other support, for example, iMatrix 511, which is a laminin 511. And we are now checking if other support also works or not.
Thank you. Britta, another question that has come in is, a recent article showed that it is very likely MS is a complication of the Epstein-Barr virus. So the individual is wondering if that's something your group is taking into account with future studies or in modeling of MS.
Yeah, so thank you for this question. This is, of course, absolutely exciting in the general MS field, and it would be indirectly tackled by us because, I mean, EBV has a B-cell trophism and not a known endothelial or brain barrier trophism to this end. And so the only context we would tackle that in, our research project, if we could investigate infected B cells in their interaction with the blood-brain barrier, or if when we have our MS-derived brain barrier models, if they alter or promote infected B-cell trafficking across the barrier. So it would be an indirect approach to this, but not a direct approach to the infection and the theory how EBV may trigger MS, maybe via molecular mimicry. Yeah.
Thank you. This question actually is for both of you, and I'll invite Hideki to answer first. What do you see as some of the next steps for this model system and some potential future applications? For example, would you suggest your model for blood-brain barrier drug delivery testing in vitro?
Yeah. So Hideaki, do you want to go first? Or, I mean, I can try it, just come back from the brain barrier storage conference where we discussed this. Which model is the best model for what? So, I mean, foremost, our model has been optimized for studying immune cell trafficking and to be grown under static and fluid shear conditions and allowing to really investigate the multi-step cascade, having all the adhesion receptors there and chemokines we need. What we do know already from RNA-seq studies we have undertaken, and then of course from those of the colleagues who have undertaken those from the other models, is that we don't have the full portfolio of transporters expressed in the cells as they are expressed at the blood-brain barrier in vivo. So I would think our model is not the first choice if you want to go for hardcore drug delivery. Then you go back to the other models that have a little bit this mixed phenotype, still some epithelioid characteristics, but the first one, so the first developed models might be much better to look at drug delivery because you have there the receptors expressed. And Hideaki can add to this because we have discussed this also, right?
Yes, I totally agree with you. And yes, but we still don't check the improvement of our model. For example, we didn't check if pericyte co-culture improves, for example, transporter expression or whatever. So we still need to work for this. Yes.
That is good. Actually, it's very timely that you already that you went to the conference and we're discussing that. So our next question is, and I'll leave this aside, which of you would like to answer, is how can we translate this study into a clinical setting and apply it to patient outcomes?
Okay. Yeah, maybe I can, as a preclinical researcher, go first and say, well, this is what we are calling already the first translational step. Because what we have done is we have the first blood-brain barrier models really derived from patients. So this is the first time we look at the barrier properties from the barrier from the patients. And so, I mean, how do you translate this? I mean, what we are proposing is that it has been overlooked in the MS etiology that there might be a contributing factor at the level of the blood-brain barrier in terms of a slight dysfunction, not a breakdown, but some subtle dysfunction that if other events happen that lead to MS, that this additional dysfunction at the BBB may contribute and may be decisive that one really develops the clinical disease. So this is in principle our idea, and then I let Hideaki elaborate on this.
Sorry, could you please
for the translation into the clinic?
Yeah.
So the question, right? Yeah, how we translate this into the clinic, which I said, I mean, we are translating already.
Yes. Yeah, we are translating, and yes, exciting part is that we are doing the RNA sequencing of these our models, and we are trying to identify causal genes for this MS BBB phenotype, and we are trying to use this molecule to reorganize and re-establish the blood-brain barrier and establish the BBB homeostasis. So we are now trying to find more possible target. Yes.
Thank you. Hideaki, this is actually a question for you. Given some of the advances in 3D culture systems and their ability to mimic in vivo settings, would it be possible? Do you think an application of something like a microfluidic system to your study would be something to look into?
Yes, yeah, it's very exciting for applying to the microfluidics And we are actually doing now, and in collaboration with James McGrath in the Rochester, New York. Mm-hmm. So we are very excited to translate to 3D model.
Great. This next question, actually, I think I will address it to Britta, you first. Again, fascinating talks and wonderful work by both of you. Would you advise if you'd be able to use this model to study the effects of brain tumor cells on the blood-brain barrier?
Yes, I mean, this could be used in order to see if again, the brain barrier might have an impact in tumor metastases. I mean, so far we only look at the tumor cell ability and always think this has a tropism towards a different organ, but this could of course be used if you have, in principle, after surgery, the opportunity to make stem cells from these patients and then differentiate the blood-brain barrier to see if there's a certain tropism for a specific, I mean, if it is melanoma, breast cancer, that are usually metastasizing to the CNS, right? Absolutely possible, totally novel area to explore, yes. Yep.
Great. Thank you very much. We've actually reached the end of our time today for the presentation. I would like to thank you both for a very engaging discussion. The Q&A session and the presentation as well. Again, thank you for taking your time to present to us today on your novel methodology for creation of blood-brain barrier models derived from pluripotent stem cells. Any questions that were not addressed will be answered by email when able. This presentation was sponsored by STEMCELL Technologies in partnership with Current Protocols. There will be an on-demand version available for viewing on the Current Protocols website at www.currentprotocols.com in the virtual seminar section under resources. Thank you everyone for attending, and again, thank you to Dr. Engelhardt and Dr. Nishihara for their time and their participation today.
Thank you very much.
Thank you.